Welcome students, in this lesson we will be learning about the buffers. We know that the buffers are the solution which are able to resist the pH change. So the substances which can resist the pH change is known as the buffer. So we can say acid-base balancing solution is known as the buffers. Actually, how it will be functioning? Buffer act as the H plus ion reservoir. It will be acting as the H plus ion reservoir. How means whenever the pH is increasing, it will be adding H plus, and as a result the pH will be decreased. Whenever the pH is going down, it will be just taking H plus, and as a result the H plus ion concentration is decreased and the pH will become normal back. So in this way the buffer is acting as a reservoir, and thus it is able to resist the pH change of a solution. Okay, now we can see that one of the best example for the buffer in an outside or open system is nothing but the sodium acetate. You can see here Na plus CH3 COO minus. Okay, sodium acetate is a best buffer in case of the open system. And in case of the living system, we can see that there is phosphate buffer, and there is carbonic acid or bicarbonate ion buffer. Carbonic acid and a bicarbonate ion buffer. So these two are in living system, and this can be in open system. Okay, so that means outside the body of an organism. It doesn't mean the open and closed system in the thermodynamic concept. I'm just saying that in an open system it means the outside the body, and the living system, phosphate buffer will be the carbonic acid, and the bicarbonate buffer will be the that will be playing a role which will be resisting the pH change. This buffer is important for living organism because we know that the functions of different molecules, biomolecules, will be changing with changing pH. We have learned it. We can just remember the ionization state is changing in case of the amino acid when you are changing the pH. In acidic pH it will be positively charged, in basic pH it will be negatively charged, and in the neutral pH it will be existing in the zwitterionic form. In the same way, ionization state will be varying for different molecules at different pH. In order to perform a particular function, that molecule should be in a particular pH. If the pH is changing, that molecule cannot function. So it is creating, or it is playing an important role in case of the living system. Now, in case of the buffer action, we can see that a buffer will be acting close to its pKa value. This pKa value we will be discussing in detail when we discuss the Henderson-Hasselbalch equation, that is towards the end of this particular unit. So pKa is nothing but the negative logarithm of acid dissociation constant minus log Ka. That is what actually mean by the pKa. So if you are taking sodium acetate, we can see that the pKa value is around 4.75. That means the sodium acetate can act as the buffer in a fully fledged manner at pH 4.7. So it will be working perfectly as the buffer at pH 4.7. So you have to keep in mind that buffer plays its role maximum at the pH close to pKa value. Let's see the buffer in biological system. We have already discussed that the two buffers, main buffers in biological system, that is phosphate or phosphoric acid buffer, and the another one is the carbonic acid bicarbonate ion buffer. Okay, so we know that it is playing an important role. The buffers are playing an important role in biological system because simply we can have one example, pepsin and the trypsin. We know that pepsin will be acting mainly in acidic pH. Okay, and that is to 1.5 to 2 pH. And in case of the trypsin, we can see that it will be acting in almost alkaline pH, above 7, in case of the intestine. So the same protein digesting enzyme, pepsin, trypsin, but they are acting at a different pH. So in order to act, in order to function, a particular biological molecule requires a particular pH. And if you are just putting that particular molecule in a different pH, it cannot work. So the biological system should resist the pH change. And that change is resisted with the help of buffers. And there are two main buffers, that is phosphate buffer and carbonic acid bicarbonate ion buffer. Let us see the phosphate buffer now. So it's a phosphoric acid buffer. So phosphoric acid buffer. So phosphoric acid is nothing but H3PO4. So we can see, you can draw the figure like this. P will be the double bond O, OH, OH, OH. This is actually what phosphoric acid is, and H3PO4. And the complete ionized state, that is inorganic phosphate, can be represented as the P double bond O, O minus, O minus, O minus. That means PO4 3 minus. PO4 3 minus. So let us see the dissociation of this particular thing. So we can see that H3PO4 will be giving one proton, and as a result it forms H2PO4 minus. And it will be having a dissociation constant of, that means the pKa will be 2.21. Negative logarithm of the dissociation constant of H3PO4 is 2.21. Okay. Now this particular H2PO4 minus will be further giving H plus ion out, and as a result it can form HPO4 2 minus. And this will be having a dissociation, that means pKa value, negative logarithm of the dissociation constant of 7.71. Sorry, 7.21. So 7.21 is a pKa value for this dissociation. Okay. Now this particular thing will be further dissociating by releasing one proton, and as a result PO4 3 minus will be formed. That is what I represented in this way. So this particular dissociation is actually helping the phosphoric acid to behave as the phosphate buffer. Now let us see which portion of this particular dissociation will be functioning as the best buffer. It is actually at this particular, because in this pKa value it will be always existing in an equilibrium state. That means the concentration of this one and the concentration of this one will be maintained at this particular pKa value. Okay, so we can see that we already learned that a buffer system will be actually performing well at its pKa value pH. So in this case, phosphoric acid buffer will be acting well or in a full-fledged manner at pH value that is close to the pKa value, and we can just say that a pH value of 7.4. So you may get a multiple choice question like the phosphoric acid is playing its maximum or playing its role in maximum at pH. It will be 7.4. As the pH 7.4, the phosphoric acid is playing a maximum good role. We can see that it's a good buffer in blood. If in blood any reactions, any chemical reactions happens, that is happening H plus ion, that is adding H plus ions, that is increasing the H plus ion concentration, what will happen? This H plus ions will be accepted by HPO4 2 minus, and as a result it will be forming H2PO4 minus. So the extra H plus ions that is released due to any chemical reactions that may decrease the pH of the blood will be consumed by HPO4 minus, and it will be forming H2PO4 minus. Now let us think in opposite way. If any chemical reactions occurs which is actually consuming the H plus ions and as a result of increasing the pH, that is deviating from the 7.4, what will happen? The opposite reaction will be happening. So H2PO4 will be dissociating and it will be releasing H plus ions, and this H plus ions will be compensating the H plus ions which is consumed by the reactions. So whenever there is an increase in H plus ion in blood, it will be accepted by HPO4 minus and it form H2PO4 minus. Whenever there is a decrease in H plus ion concentration, then the H2PO4 will be releasing H plus ions and as a result it will be compensated. And this is how the phosphoric acid buffer is actually playing a role in a living system. Now let us discuss about the carbonic acid and bicarbonate ion buffer. Okay, now let us see the carbonic acid and the bicarbonate buffer system. In carbonic acid and bicarbonate buffer system, we have to remember one enzyme, that is carbonic anhydrase, which is catalyzing a particular reaction in which carbon dioxide and water is combined to form carbonic acid, and then it is dissociated to form bicarbonate ions. Okay, let us see what is happening there. So there is an enzyme called the carbonic anhydrase. This carbonic anhydrase is actually catalyzing this particular reaction. Carbon dioxide combines with the water. It will be forming, it's a reversible reaction, H2CO3, and then this particular one will be dissociating by the same enzyme, carbonic anhydrase, and form HCO3 minus plus H plus. HCO3 minus plus H plus. So this particular reaction is catalyzed by the carbonic anhydrase enzyme. Okay. Now let us see from in this reaction who is actually playing as the buffer system. Actually this particular one is actually playing as the buffer system, and its pK value is close to 7.4, and therefore it will be perfectly working as a buffer system at pH 7.4. That means the pH of the blood. Okay, so it is actually helping in the maintenance of pH of the blood. Okay, let us see how it will be happening. As I told you before, if any reaction happens that are adding H plus into the blood, or that are adding or increasing the H plus concentration in the blood, what will happen? This H plus will be taken by the HCO3 minus, and as a result, what will happen? It will be forming H2CO3. Carbonic acid will be formed. So this particular thing is actually consuming the extra H plus ions that is formed as a result of any chemical reaction, which can decrease the pH. Okay, so that decrease in pH is blocked by HCO3 minus by combining with H plus ions and forming H2CO3 or the carbonic acid. Now, reverse is the case. Think about the reverse condition where H plus ions are consumed from the blood. As a result, the pH is going up. What will happen? The H2CO3 will then dissociate, and as a result, H plus ions are released, and this H plus ions will be compensating the one which is consumed by any reaction that happened in the blood which actually increased the pH. So that H plus ions will be released to compensate the lost H plus ions. So HCO3 minus also will be formed. So this back and forth reaction is actually playing a role in case of the human blood to fix the blood pH in around 7.4. Okay, that's about carbonic acid and bicarbonate buffer system. So we have discussed so far about buffers and different buffer system in living organism and all. So let us revise the topic. Don't forget to write the notes from the. Okay, in this slide we can see the notes. So the substances or acid-base balanced solution that resist the pH change is called the buffers. Buffer act as a reservoir of hydrogen ions. Buffer donate H plus to the solution when H plus is going down. H plus concentration in solution falls down. And remove H plus from the solution when H plus concentration increases. So sodium acetate is an example for buffer in open system. And the phosphoric acid buffer or the carbonic acid bicarbonate ion buffer is actually an example, are examples for the buffer in living systems. A buffer will be functioning best at a pH close to the pKa value of their acid component. pKa is the negative logarithm of acid solution. Okay, acid dissociation constant. pKa value is the negative logarithm of the acid dissociation constant Ka or equilibrium constant. This we can say that the sodium acetate buffer will be performing most effectively at pH around 4.7 because its pKa value is 4.75. The pKa value of the acid component, that is CH3CO minus, is actually 4.75. So buffer in biological system. In biological system, buffer plays an important role because a significant change in pH can disrupt the molecular structures of biomolecules and initiate harmful reactions. In order to avoid such negative impacts of pH change, following two buffers are playing role. First one is the phosphate buffer and second one is the carbonic acid and bicarbonate buffer system. First of all, phosphoric acid buffer system, that is phosphate buffer system. The phosphoric acid with pKa of 7.21 buffering range is an important intracellular buffer in all biosystems with a physiological pH 7.4. H3PO4, the phosphoric acid, will be dissociating by releasing H plus ions into H2PO4 minus at a pKa of 2.12, and H plus, it will be again releasing H plus at pKa of 7.21 into HPO4 2 minus. Then it will be releasing one more H plus to form the PO4 3 minus with a pKa of 12.67. So at about the pH 7.4, inorganic phosphate exists primarily as a nearly equal mixture of H2PO4 minus and HPO4 2 minus with a pKa of 7.21. When H plus concentration increases, HPO4 2 minus accept it and changes to H2PO4 minus, and when H plus concentration decreases, H2PO4 minus will be donating H plus and changes to HPO4 2 minus, acting as the effective buffer. The concentration of inorganic phosphate in human blood is about 1 millimolar, by which it can act as an effective buffer against all the processes that produces either acid or base. Now carbonic acid and bicarbonate buffer system. Carbonic acid and bicarbonate buffer system is a key buffer in human blood. Carbonic acid and bicarbonate ion interact with a pair of reversible reactions catalyzed by the enzyme carbonic anhydrase as follows. So CO2 combines with H2O. In presence of the enzyme carbonic anhydrase, it will be forming H2CO3, and then it will be dissociating into H plus and HCO3 minus with the same enzyme action. If acid is added to the blood, what will happen? HCO3 minus act as the base which remove excess H plus ions by forming H2CO3. If base is added, it will be removing H plus ions. As a result, we can see that the H2CO3 will be behaving like acid that dissociate to release H plus ions. The released H plus compensates the H plus taken by the base. Thus, the forward and reverse reactions that interconvert H2CO3 and HCO3 minus stabilize the blood pH at a 7.4.